Answering MCAT questions in under 60 seconds. Researchers are investigating the metabolic effects of a high-protein diet in two groups of individuals, endurance athletes and sedentary controls. They measure urinary levels of 1-methylhistidine, a compound derived from the breakdown of actin and myosin. Which of the following findings would best support increased skeletal muscle protein turnover in the endurance athletes? A says endurance athletes show higher serum levels of alanine transaminase compared to sedentary controls. Well, alanine transaminase is ALT, right? And there's also another molecule called AST, okay? And so ALT and AST, right, are both found in muscle, okay? But they're also found in the liver. And while ALT levels increase could indicate some elevation in muscle turnover, muscle breakdown, primarily you would expect an elevation of AST because there's a lot more AST than ALT in muscle, right? Whereas higher ALT levels are usually suggestive of liver breakdown, or liver damage, liver cell turnover, okay? So it could be correct, but we'll see if there's a better answer, okay? And then B, endurance athletes have significantly higher urinary excretion of 1-methylhistidine compared to sedentary controls. Well, they're saying that 1-methylhistidine is a compound derived from breakdown of actin and myosin. And remember, what muscle, skeletal muscle, is mostly composed of. It's mostly composed of muscle proteins, which happen to be actin and myosin. So we would definitively expect much higher levels of this metabolite, right, in muscle, in people that have higher levels of muscle breakdown compared to those that don't, okay? So that's a much better answer than A, right? That sounds like a much more specific answer. Because remember that, again, ALT is mostly going to be found in liver cells, not muscle cells. It can be found in muscle cells, but it's primarily a liver marker, okay? And then C, sedentary controls have higher circulating levels of branched-chain amino acids than endurance athletes, right? Well, branched-chain amino acids, for one thing, you know, they're not really specific to muscle cells. You know, they're used, of course, to synthesize muscle proteins, and really any proteins, but you can get these from the diet and stuff like that. That's not really specific, okay? B is still a much better answer. And then D, sedentary controls show increased urea concentration in the blood relative to endurance athletes. Well, for one thing, urea is really mostly just a marker of nitrogenous waste, right? It's how your body tries to get rid of it. And again, that could be due to, breakdown of muscle, and you have basically breakdown of proteins where you have nitrogen and whatnot. But again, for one thing, they're saying the sedentary controls for some reason have increased urea concentration, which would be probably indicative of more nitrogenous waste being produced. So it's not really obvious why that would be the case, and it doesn't tell us anything specifically about muscle breakdown. So again, B remains our best answer choice.